Hello everyone, today we will be discussing the synthesis of a thyroid hormone. The synthesis of a thyroid hormone occur in a thyroid gland. The thyroid gland is a gland situated in the neck. It consists of primarily two types of a secreting cell. The first one is a follicle or the acinus and the second one is a parafollicular cell. The follicle or acinus secrete a thyroid hormone T3 and T4 while the parafollicular cells secrete a calcitonin. The follicle or the acinus which synthesize a thyroid hormone consists of a simple epithelium which enclosed a cavity called as a follicular cavity. The follicular cavity consists of a gel-like viscous substance which consists of protein and iodine called as a colloid. You guys can see in this diagram that these surrounding cells are the simple epithelial cell and the central cavity is called as a follicular cavity which consists of a colloid. To synthesize the thyroid hormone, the follicle required two things. The first one is an amino acid tyrosine and the second one is iodine. So how it is produced and how these both are incorporated, let's study the synthesis of a thyroid hormone. For that, let's zoom the epithelial cell and a portion of a colloid. Now this side is a epithelial cell and part is a colloid. The synthesis of a thyroid hormone is stimulated by TSH. The TSH will act on the follicular cell and the follicular cell in response to the TSH will produce a thyroglobulin. A thyroglobulin is a protein which primarily consists of amino acid tyrosine. It is produced by the follicular cell. It will be packaged and will be extruded into the follicular lumen. So the synthesis of a thyroglobulin is the first step in the synthesis of a thyroid hormone. After the synthesis of a thyroglobulin, the next thing which is important for the synthesis of a thyroid hormone is iodine. So how the iodine will make entry into the follicular cell till the colloid? It begins by the active transport of iodide through the sodium iodide co-transporter in the follicular cell. The iodide will get entry into the follicular cell with the help of a sodium iodide co-transporter. After in the follicular cell, the iodide will convert it into an iodine by the peroxidase enzyme. This iodine is now a reactive form and it will undergo organification. Before organification, this iodine will get into the colloid by the pendrin. A pendrin is a channel through which the iodine will get entry into the colloid. At the junction of this follicular and the colloid, there will be an organification of the iodine, mean that the iodine will combine with the tyrosine. This is called as an organification. So to some tyrosine there will be a one iodine attached. It is called as a monoiodothyroxine, and to the some tyrosine there will be a two iodine attached. It is called as a diiodothyroxine. This formation of the DIT and MIT is called as organification. After organification, there will be a coupling. In coupling, there will be a combination of DIT and MIT. If the MIT and the DIT combine, it will form a T3. But if the DIT and the DIT combine, it will produce a T4. Generally, in the coupling, there are more T4 production than T3, but the T3 form is more active. After the organification, the thyroglobulin molecule will look like this. At some point, there will be a formation of a T3. At some point, there will be a formation of a T4, while some thyroglobulin will have a MIT and DIT which didn't undergo the organification. So this iodinated thyroglobulin will be stored in a colloid until and unless the thyroid gland undergoes some stimulation from a TSH to secrete a thyroid hormone. So whenever there is a stimulation of the thyroid cell by the TSH, there will be an endocytosis of the iodinated thyroglobulin back into the follicular cell, where this iodinated thyroglobulin will be digested by the lysosomal enzyme to produce a T4 and the T3. The T4 and T3 will go into the peripheral circulation, while the DIT and MIT will be deiodinated by an enzyme thyroid deiodinase, which will recycle the iodine, while the T4 and T3 which has reached the peripheral circulation will bind to the circulating hormone, the thyroxine binding globulin, and it will circulate in the peripheral circulation with the by binding to the thyroid binding globulin. About 99% of the T3 and the T4 are bounded to the thyroid binding globulin, and the 1% of the T4 will circulate unbound to the TBG, called as a free T4. This free T4, before get metabolically active, will convert itself into a T3, which is a more potent form by an enzyme 5' deiodinase. It was all about the synthesis of a thyroid hormone. Now let's study the regulation of a thyroid hormone. The thyroid hormone is regulated from brain. The hypothalamus release a thyroid releasing hormone TRH. The TRH will act on the anterior pituitary. The anterior pituitary will release a thyrotrophin TSH. This TSH will act on the thyroid gland, which will release a T3 and the T4. This T3 and the T4 will circulate in the peripheral circulation, and when a body requirement are met, it will inhibit the anterior pituitary and hypothalamus, decreasing the level of a TRH and the TSH. So it was all about the synthesis of a thyroid hormone. Is there still any question you guys can ask?